necessary activities
Where
right now.
Yeah. Where do you think, though, the sediment is coming from? Is that just coming off of...
Yeah, sediment's coming
from
from
setting
our
down in...
front porch that we picked up.
Science,
But not in the bio boxes, is it?
No, not from the bio boxes, just from the plates in the front.
Okay, just making sure.
Okay.
So the last I wrote, Lisa, was either 100 or 200.
Okay. 100 or 200?
I'm just checking with Lisa. That's what I last wrote down when we were reviewing it at the beginning of the dive in the science chat.
Roger.
I'm sorry. What was your question, Jen?
So they were asking about when to fire the Niskins, and at the beginning of the dive, the advice we got was either at 100 or 200 meters. But I also was having problem with my connection, so I may have missed something. So I just wanted to check in with you about.
Yeah.
Was there a final decision on that?
Darn, I'm looking at my notes. And I do not have very accurate. Darn. Sorry.
Okay.
I think I got distracted as well with something else.
Well, I think your audio was also, we were both having trouble.
Yeah. So she said it was for particulate organic matter in the water column. I don't think it mattered. Oh, here, I'm reading.
A little bit of time.
Oh, yeah. You're asking.
Yeah.
I'll send a text to, we have some time. A little bit of time.
Okay. Mm-hmm. The poem is particulate organic matter is for whom?
It's for, I think, Meredith and
300, you want to go
Nancy
start streaming?
Dowdy. So I'm sending it also to Meredith.
Okay.
Bridge nav, if you want to track a line forward at 0.3 knots. Great, thank you.
Hopefully we're not waking them up.
Oh, yeah, you're right, Kim. Kim's all over it. Or, wait.
Looks like we're holding at 374 depth for a little bit while you guys get things settled. Is that right?
To rotate.
Say that again?
We're not ascending quite yet, right?
Oh, we are currently ascending.
Yeah, we're at 285.
Oh, okay. Oh, so my Grafana is frozen. That's why I asked. Good to know.
So can Mary, can you look at the beginning of the watch change? What Meredith Everett might have said. Okay,
sure,
she's saying, I'm not sure. It's okay. Lizzie's saying, I'm not sure it would have been for the Natural History Museum or USGS
for
or
this
Taylor
last
Ann for microplastics.
This last Niskin is for USGS, I think. That's what Steve just came in and told me that. But I'll go back in the chat to double check.
Like
I have down that it was for POM and either at 100 or 200 meters.
Well,
and then my audio cut out stuff, so.
I'm going to check Nancy Bowdy's sampling protocol real quick.
Okay.
Nope.
Mhm. Yeah.
It doesn't say.
That's
Okay, so the last I see here is from
your mic.
Steve. And Steve said, we have a few POM samples from less than 200 or 250 meters. So either 100 or 200 meters is fine. That's the last I see of the discussion in the chat on that topic.
So, and I'm seeing here now areas of water column ranked according to priority: deep water, which we're past, thermocline, mixed layer, or surface water, 50 to 100 meters if the thermocline is below this range. So let's try for surface waters.
So 100? Or what do you mean?
Well, it says
What is
if between 50 and 100 if the thermocline is below this range. So let's make sure that the thermocline is below and that we're above the thermocline. That's what they're
okay? So we'll watch
yeah.
temp as we said.
We can ask Marley to put it on that one there, or that one. I guess we can go there. Hey Marley, can you put the bucket cam on one of the Argus views there?
Sorry, you guys, that this was so confusing. I totally missed it.
Oh, just the bucket cam so we can see the sponge expand.
Oh, yeah. Is it changing?
Can't really tell.
Okay.
Looks like it's still a sponge.
So we still have high pack on 3, though we have a little delay.
So science, was there consensus as to what depth we want to take this sample at? We're nearing 200 meters depth.
We're going to do it in the surface waters, so between 50 and 100 meters if we're past the thermocline.
Okay, roger
We
that.
want to be above the thermocline. Thank you.
Sure thing. So we have to stop the vehicles anyway at 50 meters, so we can do it then.
I'm about to throw the V-Link system overboard.
Sorry, Jen, we're not hearing you very well. Oh.
And I'm back. Is that better?
Yes. Yes, much better.
Great. I guess sometimes you just have to close it and reopen it. Thanks for telling me.
So yes, the sponge still looks like a sponge. It's a little crab. Is that a crab or a?
I think so.
Yeah.
Either came in on the sponge or the coral. So we're looking at the quad view. That's channel 4. And you can see inside one of the jars that's attached to the slurp, jar number 4. And in it, you can see a piece of a coral. Looks like it might be a paragorgic coral, but I can't quite tell. And that's on the left side of the view. And on the right side is a little gray and black spotted sponge. And we're looking to see if that sponge changes shape while we ascend.
And that is a paragorgid. It should be a paragorgid. We put the sponge in a jar with a paragorgid.
Great. Got that right.
Yeah.
Yay for me.
Yeah. Very good. Even with the white, very white flooding through the light that you could tell. Very good.
Well, it's going off the shape. The paragorgids seem to have more of that kind of rounded, lumpy shape. That's what I was looking at. But I am still very much learning the corals.
It's almost as if during the ascent, the Hercules gets a nice cleaning, seeing all of the debris sloughing off.
I'm curious if this watch was part of the descent when this dive began. I forget which watch it was that took the ROVs down, but yesterday I was on a call discussing conditions in the California Current, which is the water mass that moves along the western coast of the United States, and we were discussing whether or not there was still a lot of pyrosomes in the water column. I was curious if
You're seeing
on
some in
this
the
dive.
hurricane now, or where?
Okay. Yeah, I was wondering if you'd seen many on the way down. We didn't see that many on the seafloor.
That's true. I think we saw them on yesterday's dive. Not on the seafloor, but in the water column we saw a few. So this dive started at midnight today. So we've been
So the 12 to 4.
at it for 20 hours.
Got it. Okay, I was thinking it might have been 24, but I was off by 4. You started while I was
launched
sleeping.
Yes, launched at midnight. It was Lizzie and Meredith were on the first watch.
Well, we had our first dive in the Central California section was up off of Half Moon Bay, which is north of Monterey Bay, south of San Francisco. We're offshore over a feature called Pioneer Canyon, and as we descended, it was at midnight. The number of small fishes in the water column was really impressive and really fun to see. And folks had been saying that earlier they had seen humpback whales and birds in the area, and so it's kind of fun to see some of the food they were likely eating.
Yeah.
As we were going down through the water column.
So you call that very productive waters with that amount of food in it. Food from many different species.
Mm-hmm.
So tomorrow, we will move from the Channel Islands. We will move north to the Santa. Yeah, it seems like my D-Link is somewhat unstable as well, but we'll be moving to the Santa Lucia or Santa Lucia Bank area. This is a bank that rises to and we'll be looking at deeper areas of the bank. We're going to be diving at 1,500 meters to about 1,200 meters, and we'll be going to an area that has never been. And so, Jen, are you picking up everything I'm saying? Because I see that my V-Link is flipping on and off.
Yeah, I hear you a little bit. You're coming in and out. So we're having trouble
Okay.
here.
Well, there's a pyrosome right there.
Yep, there it is.
Yeah, there's a pyrosome.
Yeah,
And another.
what's the oxygen here? Because it looks pretty replete of stuff. Or deplete.
It's 30%.
Oh, okay, so it's not.
Let me
Yeah.
let
We're
me
seeing a little
refresh. Yes. No,
like
39.
siphonophore bract that was on our porch for a bit. Oh, there's a pyrosome up close. Not a huge
Yeah, not a huge
diversity of
pyrosome.
no, just not a huge diversity of gelatinous taxa. Just these pyrosomes everywhere. On the seafloor, I was seeing a lot of Keating gnats, those air worms. I don't know why I always notice those. Those are interesting to me. And then the medusa, I have a hard time IDing all the different anthomedusa and hydromedusa, but
All right.
yeah, we had a more diverse gelatinous taxa, it seemed, on near the seafloor. We only just get a little snippet as we come up. All the mud coming off. I'm curious
Ben, you
what
wanna tell Mark when we radio down to him at 50 meters that we're gonna hold for
we're gonna do a Niskin.
2 minutes to do a Niskin.
Roger.
Thank you.
Yep, almost there.
Well, oxygen's climbing. We're at 44% now.
It tends to be at those oxyclines and thermoclines.
Raj. I got the camera set up and everything though. All right. Kim, would you also mind being on the kill switch too, just in case it does weird things again?
So
Yeah.
what we're going to do right now is capture a sample of water from 50 meters below the surface in some bottles called Niskin bottles. And they're currently, we've used 5 of the 6 that we have.
I think it's the white one.
Yeah,
All right, ready.
we're going
Set
to pull this little loop.
Up.
Up.
Got it.
And the bottles were held open, and now it's basically closed and captured that bit of water that was inside. And it'll stay enclosed until it's emptied at the surface. And so it can be then analyzed, in this case, for particulate organic matter, at least that's my understanding. I don't know if we've got Lisa on the audio, and if there's other studies that are going to be done.
All right,
I just wanted to
ready.
confirm this was
Guys good?
this was sample 85.
Deck van, we're done with our Niskin. We're
Yes,
good to come
85.
up.
Thank you. And which NISC model was it? Which number?
That was Niskin number
Great.
6.
Thank you. I'm just trying to make note of that.
Definitely.
Okay.
When you're done, Lisa, based on your notes, those 6 different water samples, they're not all going to be used for the same thing, correct?
No, some of them are for eDNA. A lot of
We're losing
this one I
Lisa's
think was
audio.
for.
We heard eDNA.
Yes, a lot of them are for eDNA, and each time I talk, I lose. So
I know how frustrating it can be.
yes, it's the worst Zoom call in your life.
No, so, but the eDNA is pretty interesting. So, that's environmental DNA, and that water that was captured in the bottle will then be filtered, and the DNA that's in the water sample will be collected and then later sequenced, and we can compare the DNA that was captured in that water sample to the DNA of known marine organisms. I don't know when it cut out.
It cut out at organism. So
Oh, well, that's
tell
actually a
us
good
which
place.
organism.
It might have seemed like a complete thought.
Yeah, it was good. It was good. So when we have voucher samples of organisms that we know are in the area, we can compare and say, yes, this technique is valid, but it also allows us to get a census of organisms that live in the area that we don't usually see, like the ones that are infaunal or live in the sediment, or as you saw on the Poti Peninsula, there are many crevices and holes, and we can't see all of the animals that are living in the entire community. So this gives us a sense of the range and diversity of species that live in an area.
Exactly. And at least those are the species that have shed DNA into the water. So they do that in many ways. They lose cells like we lose skin cells, and we lose our hair. They do the same thing. They shed external components of their bodies, as well as everyone poops. So that's also being shed into the water. So waste products contain DNA. So there's various ways that animal DNA gets into the water, and that's what we're sampling when we're looking at DNA. There was some little fish that just swam through.
Yeah.
And if you switch over to the, if you switch over to either channel 2 or 4, you can see the lights of Hercules off the stern of the ship. You can see that we're very close to the surface and recovery has begun.
And it goes really fast, the recovery. I'm always so amazed how quickly it's done.
Especially when you have conditions. What I've seen off the stern looks pretty nice and flat out there.
Yes, and the way that everybody's dressed, short sleeve shirt, shorts. It must be nice and balmy down there around Channel Islands.
Yeah.
So Jen and I, we're both in north central California. I'm on the Monterey Bay, and on the north side of the bay near Santa Cruz, and Jen, you're calling in from the San Francisco East Bay area.
That's right.
Where we've also had warm weather today. Pretty hot.
Yes, it was warm.
Yeah. So that is the Argus coming on deck first, and you can see the pretty lights of the Hercules ROV, which is still in the water.
Right, and there you can see the cable that connects Argus to the ship, and all the video and all the data coming from the ROVs travels through that cable, as well as it goes through a winch, and is how Argus is raised and lowered from the ship, and then there's a yellow tether that connects Argus to Hercules. And so you're going to be seeing that next. As when they get ready, Hercules will come alongside the port side of the Nautilus, where it's recovered with a white crane. You can see someone sitting in the seat of that crane right now.
So, Jane, you've been on the Nautilus before, correct?
Yeah, 3 times. I was hoping for a 4th, but yeah.
Yeah, I was.
2017 for the Channel Islands, and then 2018 and 2019 for Monterey Bay dives. Yeah.
That's so cool.
It's really interesting to watch the choreographed dance that is deployment and recovery of these remotely operated vehicles. And it's always my favorite to see Hercules at night because it looks like a little alien, ship or something with the lights glowing around it.
I mean, the setup with the Ocean Exploration Trust is pretty phenomenal. It's, nowhere else does this setup exist. Not just with ROVs and their capability to go as deep as they can go, but with the telepresence and allowing so many other people, including us as the scientists ashore during COVID times, to still be able to explore and carry out our cruises. Many programs that do field work have not been able to do it this year. And because of the capabilities and just the wonder of telepresence, we're able to all participate as scientists ashore, but also bring it to so many people that have been watching and
following along. I know I miss having the Science Communication Fellows, which has been, I think it's another kind of unique component of the Ocean Exploration Trust program, and we definitely missed having them here to help bring to us questions from the audience in real time. So we have to now try and imagine what people want to know, but it's more fun to hear what they actually want to know, and to also hear who's watching and where they're located around the globe. It's always a favorite component of mine, and I do miss that, but I'm glad we get to continue to do the science and to live stream it and try to make it as interesting as we can along the way. I think there are a couple other vessels that have this 2-body ROV system, which is pretty. It's so nice to have that, Argus and Hercules working as a team. Gets us so many additional views of what's going on, to not have just the views from Hercules, but to have that second ROV in the water. But I don't know if those programs are running right now. Those vessels. Right. Ooh, look at all the fish right now. Maybe attracted by the lights.
would those be the vertical migrators, or would those just be So like lanternfish and ictophids, or would they just be maybe anchovies or sardines?
I mean, this is definitely an area that I don't know as much as I'd like about, but I'm seeing kind of a very silvery long body shape that makes me think more. Oh, I think I just saw some a good operculum open, like they had gill rakers, so that's making me think anchovy or sardine.
Yeah.
But I'm not getting a very good view of them. So at least you want to get a little preview tomorrow? For anyone who's still
that's
stuck
what I was
with
trying
us
to do.
during recovery? Oh, and you got cut off. That's right. Well, so far you sound really
Yeah.
good right now.
Okay. Yeah, so tomorrow we're going to the Santa Lucia Bank area, which is north of where we are right now. It's a bank that is situated about 20 miles offshore of Morro Bay, and people don't know where Morro Bay is. County.
Okay, I think
And
we
say that
lost you
again?
just for a smidge there. We lost you just
Oh.
for a smidge. So you said Morro Bay.
Okay. Morro Bay, situated in San Luis Obispo County. And this bank, Santa Lucia, has been a bank of much interest in the last 5 years, I'd say. One, because there is the Northern Chumash Tribal Council has nominated this area to be a national marine sanctuary. So it's called the Chumash Heritage National Marine Sanctuary. It's a nomination. And one day it could be selected for designation, which we hope would happen one of these days. That's a decision that is made at a very high level at NOAA, and even at the Department of Commerce level. But this nomination covers the Santa Lucia Bank, Arguello Canyon, which the Nautilus explored a couple years ago. And the area that we will be visiting tomorrow is an area that has never been visually surveyed before, as far as we know. It's a ridge that runs from about 1,500 meters to 1,200 meters. And we'll hopefully find lots of corals and sponges that have never been collected at this depth.
That sounds exciting.
Yeah.
And it's neat because it'll be a different depth than we've been looking at on these last dives. I think what was the deepest, I think, in Channel Islands was maybe at the shallower depths of what you were just describing. So this will be going a little deeper than these last two dives. So that'll be really fun to see.
Yeah, and then
I'm
so
excited
planning for these dives, I because it's so deep, we don't have any habitat suitability models for it yet. So the PODI habitat suitability models don't go that deep. And we rely a lot on mapping data to figure out the bathymetry, so the